All right, everyone, welcome. Today, we are jumping into a topic that shows up on the CUET biology exam again and again, transcription. Seriously, if you can nail this, you are paving the way for some really crucial marks. We're going to break this whole thing down, and I'll be pointing out the exact spots where students usually mess up and where examiners love to lay their little traps. So get your notes ready, stay with me right to the very end, and let's make sure these concepts are locked in for good. Okay, let's get right to it with the main puzzle. Think about this. Your DNA is the master blueprint for you, but it's locked away for safekeeping inside the cell's nucleus. It can't leave, ever. Meanwhile, the factories that build everything, the ribosomes, are way out in the cytoplasm. So how in the world do the instructions get from the protected blueprint to the factory floor? This single question is what transcription is all about, and it's the foundation for so many of the conceptual questions you're going to see on your exam. So let's use an analogy. Imagine the nucleus is like a super high-security library, and inside there's just one absolutely priceless master recipe book. That's your DNA. You can't check it out. You can't even tear a page out. It's way too important to risk any damage. So if you need just one recipe from that book, what's the smartest thing you can do? You'd make a photocopy, right? And that is exactly what transcription is. The cell creates a handy temporary copy of one specific gene. Now, this copy isn't DNA. It's a different molecule called RNA. That's ribonucleic acid. This little RNA molecule is the messenger, the authorized copy that gets a hall pass to leave the library and carry those vital instructions out to the protein factories. Now, the cell's copying machine, an awesome little enzyme called RNA polymerase, can't just start copying wherever it wants. That would be chaos. It has to know exactly which gene to copy and where that gene starts and stops. To do that, it needs a really clear map. And the structure of this map, which we call a transcription unit, is a huge favorite for diagram-based questions on the CUET. So let's break it down. Okay, this is so important. A gene is defined by these three parts. First up, you've got the promoter. Just think of it as a giant start here sign. It's where the RNA polymerase latches on. Second, you have the structural gene. This is the actual recipe itself, the part that gets copied. And third, there's the terminator, which is basically the stop sign that tells the enzyme its job is finished. Now here's a memory trick and a classic CUE trap, so listen up. To remember the order, just think pro-stit-ter, promoter, structural, terminator. And write this down. The promoter is the binding site, but it does not get transcribed. So many students lose marks on that one point. Don't be one of them. All right, this next part is something that trips a lot of people up, so let's clear it up right now. We know DNA is a double helix, right? It has two strands, but the RNA polymerase only reads one of those strands. The strand it reads is called the template strand. The other one, the one that's just kind of hanging out, is called the coding strand. Now here's the tricky part, the part the examiners love. The new RNA molecule's sequence is going to be almost identical to the coding strand, the one it didn't even read. The only tiny difference is that RNA uses a base called uracil, or U, wherever you'd see a thymine or T in DNA. Okay, so the cell made its RNA copy. Great. But, it's not quite that simple. The cell actually makes a few different kinds of RNA. You can think of them like a specialized factory crew, where each member has a very specific and important job to do to get that protein built. And here are the three key players you absolutely have to know for CUET. First, there's mRNA, the messenger RNA. The name says it all. It carries the genetic message. Second is tRNA, the transfer RNA. It's like a little tow truck transferring the right amino acid building blocks over to the ribosome. And third is rRNA, or ribosomal RNA. This one is actually a physical part of the ribosome factory. A super simple memory trick, just remember MRT for messenger, ribosomal, and transfer RNA. Now, for complex cells like ours, eukaryotes, the story doesn't end there. In simpler cells like bacteria, that mRNA copy is ready to go right away. But in our cells, that first copy is really just a rough draft. It needs a good bit of editing and polishing before it can be sent out. This whole stage is called RNA processing, and I can almost guarantee you'll get a question about the sequence of its steps. This initial raw RNA transcript has a special name. hnRNA. That stands for heterogeneous nuclear RNA. You can just think of it as pre-mRNA. It's full of extra bits of information that aren't actually needed to build the protein. And remember this key difference, because it's a huge source of comparison questions. This whole fancy editing process only happens in eukaryotes. Prokaryotes, like bacteria, don't have a nucleus, so their process is way more direct. So what exactly gets edited out? Well, the hnRNA has two types of sequences. The parts that need to go are called introns. I want you to think of introns as the junk mail in your email inbox. They're just in the way. The important parts we need to keep are the exons. They are the expressed sequences that have the actual message, and the process of cutting out all the introns and stitching the exons together is called splicing. A common mistake here is thinking introns are totally useless. They don't code for the protein, sure, but they can have other jobs. But for this final message, they have to go. And here it is, the full editing checklist. The order here is absolutely critical for your exam, so pay attention. First, at the very front of the RNA, the 5' end, a protective cap is added. We call this capping. Second, at the back end, the 3' end, a long tail of adenine bases gets added. This is called a poly-A tail, or tailing. Finally, the introns get cut out and the exons get joined together. That's splicing. So how do you remember the order? It's easy. Cap, tail, cut. Put on the cap, add the tail, then cut out the junk. Simple as that. All right, here's a question to get you thinking conceptually, just like the CUET exam will force you to. Pause for a second and really think about it. Why do complex cells like ours have this whole complicated editing process, while simple bacteria just don't? Well, the answer is all about their cell structure. Bacteria don't have a nucleus. For them, transcription and the next step, translation, are happening at the same time in the same place. There's literally no time or space for all this editing. It's all about speed and efficiency for them. So, we've been deep in the molecular details, I know, but let's zoom out for a second. Why does all this matter? Why is transcription such a foundational topic? It's because this process is the direct, living link between your genes, the static, written code in your DNA, and the dynamic, functioning reality of who you are, which is all built out of proteins. And its importance is everywhere you look. Those revolutionary mRNA vaccines that helped fight COVID-19, they work by hijacking this exact system. They give our cells a little piece of mRNA to read so they can build a defense. A lot of devastating genetic disorders, like some types of muscular dystrophy, are caused by just one tiny mistake in splicing. And cancer, at its core, is a disease of transcription gone haywire, where genes get turned on or off when they absolutely shouldn't be. Understanding this process is central to all of modern medicine. Now, let's take an even bigger step back, all the way back to the origin of life itself. We find RNA right there, center stage. This is a hot topic for those assertion-reason questions in CUET. There's this idea called the RNA World Hypothesis. It suggests that before life decided to use stable DNA for storage and versatile proteins for doing all the work, there was an age of RNA. Early life might have been based entirely on this one molecule. So why RNA? What's so special about it? This quote just nails it perfectly. RNA has this unique, two-in-one ability that makes it the perfect candidate for kickstarting life. It can store genetic info, just like DNA, and it can fold itself into complex shapes and act like an enzyme, speeding up chemical reactions, a job mostly done by proteins now. These special catalytic RNA molecules are called ribozymes. The fact that it can be both the blueprint and the builder makes RNA the ultimate multi-tool of biology. That is a very, very important concept for your exam. Okay, let's do a super fast revision, and then I'll test you. Key takeaways: the transcription unit is pros-T-ter. The enzyme is RNA polymerase. The template strand gets read, but the RNA copy looks like the coding strand. Eukaryotes have processing: cap, tail, cut. Prokaryotes don't. Ready? Let's try a question. Which of the following is removed from hnRNA during processing? A, exons, B, introns, C, promoter, or D, terminator? Pause and think. The answer is B, introns. Exons are expressed, and the promoter and terminator aren't even in the transcript. Good. One more, a tougher one. In a transcription unit, the promoter is located towards which end of the structural gene? A, 3' end of the template strand, B, 5' end of the template strand, C, 3' end of the coding strand, or D, 5' end of the coding strand? The correct answer is D, the 5' end of the coding strand. That defines the upstream region. This incredible, precise process happens constantly in your body, and as we saw, a single typo can lead to disease. It's a powerful reminder of how finely tuned this whole system is. And that's a wrap on transcription. If this helped, join our community of serious CUET aspirants and hit that subscribe button. Next time, we're tackling what happens next, translation. See you then.